So there's a really great review in Nature Protocols a number of years back, looking at some of the common cell potency assays that are used in the field. So there's 5 that I'm going to go over in pretty limited detail, but if you do want to know more, definitely go look up this review. So the first one is the chromium release assay. This is a well-established protocol in the field. It does use chromium to label the target cells, and then you co-incubate your effector cells for various amounts of time, and then the target cells, if they are killed, release chromium, and you can detect that in the plates. So this has a low throughput, but is very sensitive, but does require radioactive materials. So it's not as common and prevalent as it once was because of these new and upcoming technologies. One of the ones that we're going to actually show you data on today is the bioluminescence assay. So this is where the target cells have been genetically modified to express a reporter gene. In this case, we're using luciferase. So the target cells, if they're killed, are not there to have any luciferase left, but the target cells that are not killed, when you add the luciferin enzyme, can bioluminesce. So that way you can assess really quickly through high throughput means whether or not there is specific killing. The next one is an impedance assay. So this is a cell detachment assay where the target cells are attached to the bottom of the plate. And if they are killed, they are released off the bottom of the plate. So this is the only one that can have a real-time measurement and is also nice and high throughput. The second to last one is a flow cytometry assay. So this is, once again, co-culturing those cells together and looking for the presence of annexin 5 and 7A-AD on those cells. So those are molecules commonly associated with dead and dying. So annexin AAV being a phospholipid that gets flipped, and then you have staining of the nucleic acids inside the cell. So once again, can be pretty high throughput. The final one that we're going to talk about today and is most relevant to the talk is the interferon gamma release assay. So like all the other assays, you're co-culturing those cells together, but in this case, you are harvesting the supergenes at the end, and you are running an ELISA to be able to detect the presence of interferon gamma that would have been secreted by the effector cells. So in its current kind of standard form, an ELISA is pretty low throughput, but we will be talking about today how we are generating some much more high throughput, better assays for the future of this type of potency assay.
Thank you for watching this webinar clip from Cell and Gene Therapy Insights. Follow the link in the description if you'd like to see the full webinar. For more clips like this, subscribe to our channel, and for more content covering the latest news, trends, issues and breakthroughs, check out our journal webpage using the link in the description.